Today again I'm back with you to talk a little more about genetics. Here in this, again I have brought forward a series of lectures where we will try to learn what is gene or what makes up genes, how do genes work in a body, or in an individual, then how these genes, we have all this time we have been learning that genes, they pass on from one person to one generation to the next generation and so on and so forth. But then what actually passes on in the form of a gene? What makes up genes? How do they express the Mendelian characters that we talked about earlier? So all of these we will try to understand, we will try to learn together in this series of lectures. So here I start with the first of it. Now before moving forward, okay, let us go to some basic concepts. First is, what is cell? Cell, as we all know, is the structural and functional unit of life. What makes up cells? Cells are made up of a mass of protoplasm bounded by cytoplasm and containing a nucleus with nuclear material in case of eukaryotes, and in case of prokaryotes the nuclear material is present but it will not remain confined within the membrane. So cell contains nucleus in case of eukaryotes. What is the nucleus or the nuclear material made up of? It is made up of chromosomes that we have always been talking about. The number of chromosomes marks the identity of a species and so on. So now what makes up the chromosomes again? Chromosomes are made up of nucleic acids. Okay, they're made up of nucleic acids. Nucleic acids which are again of two types, ribonucleic acid and deoxyribonucleic acid. Nucleic acids are made up of polynucleotide chains. Now the ribonucleic acid, deoxyribonucleic acid, both of them are present within the nucleus in eukaryotes and as a part of the nuclear component in case of prokaryotes. Both of these two. Now the basic functions of the nucleic acids. Ribonucleic acid, they act as messengers, they act as adapters, they act as catalysts, biocatalysts. About all of these we will be learning in the coming lectures. The details we will be learning about them. Then deoxyribonucleic acid, what is the main function? It is that it acts as a genetic material for most of the organisms. It is this material that is being transmitted or that is being transferred from one generation to the next. Now little more, the cell, this is the cell. Okay, what I've told just now is the cell. Cell has the nucleus, so it is the nucleus. Nucleus has the chromosomes. You can see these are the chromosomes, and these chromosomes are made up of the nucleic acid. And nucleic acid, as here, as already told, mostly the genetic material is deoxyribonucleic acid. So in most cases, okay, or rather almost all cases, the chromosomes are made up of nucleic acid, deoxyribonucleic acid. The structure of it appears in this manner. So these are some basic things which we have to keep in mind in order to get the concepts of the next part. Now polynucleotide chain. Okay, what are polynucleotide chains? Polynucleotide chains are the polymers, are the biomacromolecules found as a part of the nuclear component, enclosed within the membrane in case of nuclear membrane in case of eukaryotes, and not enclosed within a membrane in case of prokaryotes. They simply lie within the cytoplasm. Now the polynucleotides, as I told you, they are polymers. They are biomacromolecules. They are made up of repeating units of nucleotides. Now nucleotides, again in turn, they are made up of three components. Okay, phosphates. Okay, nucleotides, they are made up of three components, that is phosphates. Here you can get the structure of phosphate, phosphoric acid or phosphates. They're made up of pentose sugar. Okay, pentose sugar. Now we have to focus on this mainly. Pentose sugar, that is a 5-carbon compound. A 5-carbon compound. Here the carbons, they are marked. They are marked. Here we have the ribose structure and this is the deoxyribose structure. So the sugar, it can be ribose sugar or it can be deoxyribose sugar. So ribose sugar, okay, all the carbons that are present, okay, they are the valencies, they are fully occupied, okay, they are fulfilled, the positions, so it is complete. Now deoxyribose sugar, what happens, the second carbon, the second position carbon is devoid or is deficient of oxygen. You can see here OH, here it's O is missing, okay, it's devoid of oxygen and hence the name 2-deoxyribose. 2 means second position, deoxy, deficient in oxygen. So this is the second component that makes up one nucleotide. Then the third component is a nitrogenous base. Nitrogenous base means a compound which is quite rich in nitrogen. There are two types of nitrogenous bases, mainly purine and pyrimidine. So one nucleotide will be made up of phosphate, pentose sugar and either purine or pyrimidine. One of them will be present to form a nucleotide. Now purines and pyrimidines can again be of different types, like say purines, they are of two types mainly, adenine and guanine. Pyrimidines are mainly of they are either thymine, cytosine or uracil. So in total we have, okay, or in total in the living system there are 5 types of nitrogenous bases, 2 purines and 3 pyrimidines. But for polynucleotide chains, if it is a deoxyribose, then only 4 types will be present, that is adenine, guanine, thymine and cytosine. If it is ribonucleic acid, then also only 4 types will be present, that is adenine, guanine, cytosine and uracil. So what we can infer? We can infer that adenine, guanine and cytosine are common for both deoxyribose and ribose. Thymine, uracil, they are not common. Thymine is present in ribose, deoxyribose, and uracil is present in ribonucleic acid. Okay, so these 3 components, phosphate, pentose sugar, and nitrogenous bases, together makes up one nucleotide. Such nucleotides, they join with each other to form a polynucleotide. Now, the structure, how it is formed. Sugar joins up, adds up with or forms a bond, to be more particular. Sugar, that is the pentose sugar, it forms a glycosidic bond with the nitrogenous base, which can be either a purine or a pyrimidine. That means it can be either adenine, guanine, cytosine, thymine or uracil, any one of them. It forms a glycosidic bond at the second position of sugar, second position. Okay, whether it's deoxyribose or ribose, whatever be it, it will, that the first position of the sugar will form a glycosidic bond with the nitrogenous base, a particular nitrogenous base. That structure will be called as nucleoside. Sugar, the first carbon forming glycosidic bond with a nitrogenous base. Together, these two referred to as nucleoside. Nucleoside then at the 5th position of carbon, okay, because here carbon is intact, okay, carbon has only the base at the first position, then the 5th position, 5th sugar will attach with it, okay, here you can see here nucleoside base plus sugar, okay, then it will get attached with a phosphate, okay, it will get attached with a phosphate at the 5th position to form a nucleotide. Here you can see, okay, this is the sugar, okay, it attaches with the base, blue is base, green is sugar, okay, then this is the first position that they have shown here, and at the 5th position phosphate joins to form a nucleotide. So nucleotide, okay, here again we get the same structure. So here the 5 carbon sugar, the first carbon linking with the N-glycosidic bond, this bond name of the bond, with the nitrogenous base, which can be either of these. Here they have shown deoxyribose, that is why here uracil is missing. So at the 5th position, phosphate group. This is one nucleotide. Then when two such structures join together, now how will they join together? Now here you can see the third carbon. Third carbon is free. Now as this is the first nucleotide, okay, takes its position. Second nucleotide comes here, okay, comes to lie just behind it or ahead of it, wherever be it lies. And then the nucleotide, it has the phosphate here. It has the phosphate. Here also the nucleotides, phosphate is present here. Now the third position of the nucleotide lying ahead, the third carbon will form an ester bond with the phosphate of the next nucleotide. You can see here, this one. This is the ester bond. And also, before that also, the 5th carbon always gets added or attached to the phosphate with the help of an ester bond. So here what do you see? We see 2 ester bonds. Hence, this linkage is known as phosphodiester bond. Then polynucleotides. So this way, when they go to form one after another, first nucleotide, second nucleotide, third nucleotide. Getting attached, first nucleotide, the phosphate lies here. Second nucleotide, the phosphate lies here. The phosphate of the second nucleotide getting bonded with the third carbon of the first nucleotide. The phosphate of the third nucleotide getting linked with the carbon of the second nucleotide. So it goes on getting linked one after another. Thereby it forms a polynucleotide chain. And the bases, what will happen? The 4 types of bases, they will get bonded here randomly. Again, polynucleotide structure. Okay, the molecular structure, it is shown here. As you can see here, this is a deoxyribose. Okay, here one oxygen is missing here. So the phosphate, here's the phosphate of the first nucleotide. Okay, if I go in this direction, so this is the 3' end. This end is considered as the 3' end, which is not bonded to any phosphate. And obviously the other side where we have the free phosphate, this will be called as the 5' end. So we always mark the polarities 5 to 3, 3 to 5. Okay, we mark the polarities of a polynucleotide chain. So a recapitulation, what we learned: nucleic acids are present inside the cells. Nucleic acids are of 2 types: RNA, DNA, ribonucleic acid, deoxyribonucleic acid. They are both made up of polynucleotides. Polynucleotides are made up of nucleotides. Nucleotides in turn are made up of nucleosides and phosphate. Nucleosides are pentose sugar and nitrogenous bases, plus phosphate together makes up nucleotide. Then the types of bonds that are present, that is glycosidic bond, which will help in attachment of pentose sugar with nitrogenous base, and phosphodiester linkages, which helps in attachment of the phosphate. Okay, then free OH at the 5' end, and the other end has free OH at 3' end. Okay, so the 5' end and 3' end. Then sugar phosphate, okay, sugar phosphate, which are common, okay, which are common for all nucleic acids. Okay, how does nucleic acid vary from one to the other? Because there is difference in the bases. Otherwise sugar and phosphate is common. Hence sugar phosphate is called as the backbone. Now we will continue into our second lecture.